52 years of endless hard work, of prolonged dedications, of persistent efforts, for the best experiences, for advanced knowledge, for outstanding researches, achievements to create a better future.
Jember berada di tengah-tengah di jantung wilayah perkebunan. Area perkebunan ini memberikan bahan-bahan yang luar biasa kaya untuk bisa meneliti mengenai pengetahuan masa silam. Itu saya kira jadi alasan penting bahwa sumber sejarah itu bisa dikumpulkan di sekitar Universitas Jember.
budaya Pendalungan ini merupakan budaya baru yang saya belum pernah ketemu, dan saya bisa ketemu budaya itu hanya di Jember.
It's the whole thing because I think our Asia is so interesting and it's a complete difference to Europe, and I was very interesting, or I'm very interesting in the culture and the religion and how it affects the life here, the life and the university.
Kona ba pariwisata, hau sangat konten ya Jember, tambah yane hau sering lau halimar ba pantai-pantai ni Jember. Depois nya wisata mos termasuk diak dibandingin husi paten sersi seluk. Poi janim hal hau tertarik nomos hakarak halau studi na pating lanjut ya Universitas dari Jember bener.
Ini yang luar biasa berpotensi itu adalah karena UNED itu mempunyai akreditasi A. Kemudian semua keilmuan yang ada saat ini itu ada di Universitas Jember.
Jadi di sini saya melihat bahwa range ilmu itu sangat banyak sekali kalau orang datang ke sini, juga didukung oleh lingkungan Jember yang mungkin tidak seperti di kota-kota besar yang terlalu banyak kalau buat mahasiswa itu terlalu banyak godaan.
Pada awalnya saya tidak membayangkan keadaan Universitas Jember itu seperti apa. Namun setelah saya berkuliah di sini, persepsi saya tentang Universitas Jember jauh dari apa yang saya pikirkan. Universitas Jember memiliki banyak potensi yang mendukung potensi saya sendiri, baik dalam pendidikan maupun seni ataupun olahraga.
Kita biasanya hanya melihat dari luarnya aja, dengan banyak sharing, kemudian kuliah, juga pekerjaan di laboratorium. Di situ yang sangat memacu saya bahwa ternyata masih sedikit yang saya tahu, sehingga saya harus lebih banyak belajar lagi.
Kalau Anda ingin belajar perkebunan, kopi, kakao yang ada hanya di sekitar Universitas Jember. Tidak akan ada yang lain, karena pusat penelitian kopi dan kakao hanya ada di Jember. Satu-satunya di Indonesia. Dari segi agro ya, maka tidak ada tempat lain untuk belajar tebu kalau enggak ke Universitas Jember. Dalam hal biologi yang terkait dengan tropical resource ya, kita punya hal yang lebih lengkap, tidak sekedar daratan, tapi kita juga punya pantai, kita juga punya laut, dan kita mengeksplorasi semua itu. Dalam hal kesehatan kita berbeda. Yang lain bicara tentang patogen, kita bicara tentang vektor. Nah, jadi dari situ mestinya orang berpikir, saya tidak ada tempat yang lain untuk belajar kecuali saya datang ke sini.
Yang selama ini saya terapkan adalah konsep belajar learning by doing, atau belajar karena memang melakukan itu. Nah, jadi di dalam proses belajar mengajar itu tidak hanya sifatnya menyampaikan teori-teori tanpa melihat langsung konteks yang terjadi di lingkungan sekitar.
Teknik mengajarnya itu sangat mendewasakan untuk mahasiswa. Kita juga dipaksa untuk belajar sendiri, dan itu bisa membantu mahasiswanya juga untuk terus berkembang.
Awareness mahasiswa dari ilmu yang dia dapat di kelas, kemudian diaplikasikan di penelitian dia, kemudian sampling di lapangan, mengetahui kondisi daerah endemik malaria itu seperti ini, karena masyarakatnya belum tahu dan seterusnya. Jadi itu akan melahirkan kepedulian. Itu yang saya sebut sebagai salah satu soft skill. Dan gimana setelah peduli untuk usaha bersungguh-sungguh untuk bisa mengatasi permasalahan tersebut. Jadi memang itu sudah menjadi salah satu komitmen ya Universitas Jember untuk bisa mendidik, tidak hanya menghasilkan ilmuwan, tetapi juga ilmuwan yang juga peduli dengan kondisi masyarakat.
Nah, rasa peduli itu, saya baru sadari bahwa itu tidak bisa didapatkan di universitas. Itu merupakan suatu live university, yaitu bisa dilatih dengan cara terjun langsung ke dalam masyarakat itu sendiri, sehingga dapat menimbulkan atau
Universitas Jember adalah perguruan tinggi yang ada di timur. Tetapi ingat, prestasi kita tidak kalah dengan perguruan tinggi lainnya. Universitas Jember selalu memancarkan cahayanya dan memancarkan alumni-alumni yang berkualitas, yang mengharumkan nama Universitas Jember dan memberikan kontribusi bagi bangsa dan negara Indonesia.
Rawir sekali Universitas Jember manukana.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Headmaster of Study Program of Public Health Science, University of Jember, Head of Agomedicine Development Committee, our keynote speaker in general language, general lecture, and all happy audience. Praise be to Allah Subhanahu wa Ta'ala, who has given us mercy, guidance, happiness, and healthy, so we can attend and participate in this event without any obstacles. Praise and salutation. Praise and salutation upon to our Prophet Muhammad sallallahu alaihi wasallam, the last messenger, the best figure of this universe. The person was able to save us from the darkness to the lightness and guided us to the right pathway. In this special morning, we have several agenda of General Lecture, Study Program of Public Health Science and Biotechnology, University of Jember, held on Surahman Hall on Wednesday, 16 November 2016. So allow me to read several sequences of our agenda today. The first is opening, the second is singing Indonesia Raya, the third is greeting speech, the fourth is praying, the fifth is general lecture and discussion, the sixth is closing. Well, to advantage our quality of our time, let's start our programs today by reciting Basmalah. Bismillahirrahmanirrahim. The second agenda is singing the national song, Indonesia Raya, which will be led by Sister Dina. Time is yours. For all the audience, please stand up. For all audience, take your seat, please. The third agenda is greeting speech from Director of Postgraduate Program of Jember University, which is represented by Headmaster of Study Program of Public Health Science University of Jember. So, Dr. Isa Ma'rufi, Magister Kesehatan, time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, good morning Prof Angel. Ya. Terima kasih Bapak Ibu semua ya pada hari ini bisa menghadiri kuliah umum ya yang dilaksanakan oleh Prodi Ilmu Kesehatan Masyarakat, termasuk kemudian juga mengundang teman-teman kita di bioteknologi ya. Kebetulan pada hari ini kita kedatangan seorang ilmuwan ya dari Amerika ya, Prof Angel. Moga-moga nanti apa yang diberikan beliau ya akan sangat bermanfaat dan memotivasi tentu Bapak Ibu semuanya untuk menjadi seorang ilmuwan ya. Jadi kuliah S2 memang benar-benar dicetak Bapak Ibu untuk menjadi seorang ilmuwan. Mohon maaf disampaikan oleh Bapak Direktur ya pasca sarjana karena pada pagi hari ini ya Bapak Direktur tidak bisa memberi sambutan dan menghadiri acara ini karena ada acara yang tidak bisa ditinggalkan terkait pada tugas, sehingga pada hari ini ya saya sebagai Kaprodi Ilmu Kesehatan Masyarakat diminta untuk sedikit memberi sambutan pada kuliah umum pada hari ini. Bapak Ibu semuanya, bahwa kuliah umum hari ini merupakan agenda rutin ya yang kita laksanakan. Kita insya Allah akan melaksanakan setiap mungkin 2-3 bulan sekali untuk mengasah ya keilmuan yang terkini ya yang ada, sehingga kemudian Bapak Ibu akan mendapatkan wawasan tambahan, tidak hanya sekedar kemudian itu nanti didapatkan di bangku kuliah seperti itu. Ya, kalau kita lihat memang ilmuwan itu kan apa, orang yang ahli menguasai pengetahuan di bidang ilmu, sesuatu ilmu ya itu. Sehingga Bapak Ibu semuanya ketika nanti Bapak Ibu belajar S2 kemudian menguasai di bidang sesuatu, apakah ilmu kesehatan masyarakat atau kemudian bioteknologi, kami harapkan bahwa Bapak Ibu kemudian akan menyenangi dari ilmu pengetahuan yang Bapak Ibu kemudian pelajari seperti itu. Apa yang nanti disampaikan Prof Angel, bagaimana kemudian memotivasi Bapak Ibu agar menyenangi apa yang menjadi keahlian Bapak Ibu semuanya, seperti itu. Kita sebagai ilmuwan itu adalah sebenarnya sebagai jembatan ya, jembatan antara ilmu pengetahuan dan masyarakat. Di satu sisi ada ilmu pengetahuan ya dan teknologi, di satu sisi ada masyarakat, di mana kemudian masyarakat dalam mendapatkan ilmu pengetahuan ini juga agak sulit ya. Sehingga peran Bapak Ibu nanti adalah sebagai jembatan dalam apa, menyampaikan ilmu pengetahuan yang ada itu ke masyarakat, sehingga kemudian masyarakat itu tahu akan sesuatu, tahu akan ilmu pengetahuan. Nah, seorang ilmu pengetahuan itu tentu tidak akan mudah mengembangkan keilmuan kalau kemudian dia tidak menyukai ilmunya itu sendiri. Untuk itu memang yang pertama kita harus suka, kita pertama harus cinta terhadap ilmu itu sendiri. Ketika kita suka, ketika kita cinta, maka insya Allah kemudian kita akan menyenangi, menyukai terhadap ilmu tersebut. Nah, bagaimana kemudian nanti kita menyenangi ilmu pengetahuan, atau kita sebagai seorang ilmuwan itu kemudian menyenangi sebagai seorang ilmuwan? suka ya, itu nanti akan dibahas secara mendalam oleh Prof Angel, dan semoga nanti kuliah umum ini akan benar-benar memberikan wawasan tambahan bagi Bapak Ibu semua yang luar biasa ya terhadap sisi ilmuwan sendiri, termasuk bagaimana kemudian kita menyenangi terhadap ilmu yang kita pelajari, seperti itu. Itu mungkin sedikit sambutan dari kami ya. Saya akhiri ya. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you to Mr. Isa Marufi. The fourth agenda is praying. The fourth agenda is praying, which will be led by Brother Amal Soleh. Times is yours.
Assalamualaikum warahmatullahi wabarakatuh. Bismillahirrahmanirrahim. Alhamdulillahirabbil alamin. Hamdan yafi ni'amahu wa yukafi mazidah. Ya Rabbana lakal hamdu kama yanbaghi jalali sultanik. Allahumma ya Allah, jadikanlah acara kuliah umum ini sebagai majelis ilmu yang manfaat, guna menambah wawasan keilmuan kami, meningkatkan persaudaraan kami dalam beribadah, serta memotivasi hidup kami agar istiqomah di jalan-Mu. Melalui acara ini, bimbinglah kami untuk selalu mencari ridha-Mu, petunjuk-Mu, ilmu-ilmu yang bermanfaat bagi masyarakat di masa yang akan datang. Allahumma ya Allah, jadikanlah kami yang hadir di sini menjadi insan yang amanah. Anugerahkanlah kami tekad, kekuatan, keikhlasan, lindungan, dan bimbingan untuk menjalankan amanah sebagai insan yang bekerja dan berkarya demi mengabdi kepadamu. Allahumma ya Allah, kami yakin kami semua adalah hamba-hamba yang penuh dosa dan kesalahan. Oleh karenanya, ampunilah dosa dan kesalahan kami, orang tua kami, guru-guru kami, pahlawan bangsa kami, dan para pemimpin kami. Terimalah segala amal dan pengabdian kami dengan ridha dan rahmat-Mu. Rabbana dhalamna anfusana wa illam taghfirna wa tarhamna lakunanna minal khasirin. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar. Subhana rabbika rabbil izzati amma yasifun wa salamun alal mursalin walhamdulillahi rabbil alamin. Al-Fatihah. Assalamualaikum warahmatullahi wabarakatuh.
Thank you. And now is our main program. To all the audience, our general lecture by the topic, Why It's Fun to be a Scientist, with the keynote speaker, Mrs. Angel N. Fanajihara, PhD from University of Hawaii at Manoa, USA, is going to be started. In this lecture, we will be guided with someone which expert in biomolecular. Let us say welcome to Dr. Al-Munawir, PhD. Time is yours. And for our keynote speaker, give applause to Mrs. Angel N. Fanajihara, PhD from University of Hawaii at Manoa, USA.
Ketindian ini ya sudah ada.
Baik, assalamualaikum warahmatullahi wabarakatuh. Selamat pagi semua. hari ini kita kedatangan tamu ya, jadi kita harapkan nanti kalian semua dapat langsung ya, karena ini sangat langka sekali seorang ilmuwan mau datang ke tempat kita ya. Ini nanti kita bagi dua ya, yang pertama presentasi dari Profesor Angel dan kemudian nanti ada tanya jawab. Okay, I will to start the lecture today. I want to give you time for lecture. I think it's 1 hour. Is okay? Yeah, and then we can continue to discussion, right? Okay, please give lecture.
Good morning, and thank you for attending. It's a great honor to be here today. I'd like to give you an overview of my research over the last 19 years, and in a broader sense, it should address the question why it's fun to be a scientist. Basically, the best way for me to answer that question is through my own career path and my own career story, which, when I look at it all together, is certainly a very interesting and amazing career path. So I would say that it is fun to be a scientist. But to be quite honest about it, it isn't easy, and it's a path that requires a great dedication. There, hello? Okay, test, 1, 2, good. All right, and okay. The overview of my work is under the title Indo-Pacific Box Jellyfish Envenomations, and because I understand that you folks are students, graduate students in public health and biotechnology, I have focused the work that I'm presenting today on those items which are of relevance to you. And I would challenge any members of the audience here to consider a career to join this team effort. This is really a new chapter in box jellyfish research around the world, and Indonesia plays a pivotal role in this chapter, and we basically are starting with almost a blank slate. Can I advance the slide here, or? Oh, there, okay, thank you. In summary then today, I hope that you will learn and be challenged to answer the following questions. Are you a scientist because of educational credentials alone? That is to say, if the folks in this room complete their degree programs with a master's certificate or a PhD, does that make you a scientist? Do you want to use your degree to address human health issues and uncover new discoveries that can be the basis for novel biotechnologies? I would hope that the answer to that is yes. It's all well and good to study hard and to get good marks and to get a degree, but if there's no greater good served as far as to your fellow man at the end of the day, what was the purpose of your education? Just to have a nice degree on the wall, or to actually make a contribution to lessen human suffering around the world, and in particular in Indonesia? What does it mean to have a career in basic research? I think that that's a very complicated question, and I won't even try to really answer it. I will give you an example of my own career in biomedical research and what that has entailed. Now, moving on to the specific case which my career encompasses, I'm going to talk a little bit in detail about box jellyfish. And I know that that might not be of interest to you right now, but in terms of your ability to serve your community with regard to public health issues, it is a larger issue of public health relevance in Indonesia because there are deaths and grave injuries due to box jellyfish. So I want to give you the fundamental mechanistic basis for these kinds of injuries and deaths. So what are box jellyfish? And how can these stings lead to death in as little as 5 minutes or with a delay in up to 48 hours? What are the best preclinical and clinical care approaches? What are the current research priorities? And how can international collaborations reduce annual deaths? More and more we are living in a shrinking world in which international collaborations are absolutely key to authentic research progress. There is a great body of work that is the scientific literature, and papers are constantly being published. But whether these papers are of value to the greater good as far as serving public health need or not is not necessarily represented by the number of publications that occur. So it's really imperative on basic researchers such as myself to interact more fully with folks in public health as well as primary care physicians. If we stay in our laboratories and we don't interact, or if we publish papers which don't have public relevance, I would argue that we aren't really doing our best as what it means to be a scientist. So as far as what are box jellyfish, I hope you will see that this is the entire animal kingdom as far as biodiversity goes with regard to genetic species. When we speak about box jellyfish, they are very early in the animal kingdom, very primitive, after basically plants and fungi. Here we have the Cnidaria. This phylum Cnidaria includes box jellies, corals, anemones, etc. So cnidarians are all these animals, the sea anemones, the corals, Scyphozoa, Hydrozoa, Cubozoa, not shown here, Staurozoa. And these animals all share in common what's called cnidae, or nematocysts, or stinging cells. And these cells explosively discharge upon contact. The individual cells contain a cyst, and in the case of these penetrant cnidae, they are called nematocysts. There are many different types of cnidae. Only one class is penetrant. And here's shown this hollow tubule which is ejected at bullet speed and empties into the tissue the venom. So it pierces through the shrimp shell or the fish scales or human skin to penetrate deep in, as deep as 1 millimeter, and eject out this venom. The venom isn't a nice liquid mixture like a snake venom would be or scorpion venom. It's highly lipidic. So it actually oozes out, not quite as quickly as you see in that picture, over the course of hours. Box jellyfish also have a unique attribute in that they have eyes, such as our own, with a retina, a cornea, a lens. They have 24, oops, sorry, can we go back? Yeah. And I need you to press the play button on the embedded movie. Computer person. So if you, the black box, on the black box there's a play button. Okay. Did we lose our computer person? Yes, there, the arrow. On the black box, at the bottom of the black box. No. Oh. Can we call back the computer? There, the arrow, you see? There, the arrow, yes. Okay. And perhaps he can return to his seat. Oh, okay, thank you. So you can see now the eye, and there are 4 eye stalks. Each of these eye stalks have 6 visual apparati, 2 of them, such as our own, with a retina, a lens, iris, that respond to light as our own eyes do, with the iris constricting or dilating. Okay, thank you. So there are 2 basic families within the class Cubozoa. They are the cryptid and the chirodropid. It's not clear to me what has been the culprit in the deaths in Indonesia. We don't have good description. We went up to North Java to visit, and we met with a fisherman, Mr. Budi, who described these as olayan, and he drew a picture. But his picture, perhaps it was more like a chirodropid, but he described a very small animal. So importantly, we need to also reach out to marine scientists at Jember and basic biologists to join this community effort. So where are box jellyfish found? Basically, you can see they're around the world, throughout principally the tropics. Severe fatalities have been recorded in Australia, in the Philippines, in Thailand, throughout this Indo-Pacific region. But you'll notice that Indonesia is a blank spot on this map. We know that there have been deaths in Indonesia, but they haven't been clearly delineated as to the causative agent. So buru-buru, or jellyfish, isn't sufficiently detailed for us to be able to include that on our global epidemiological map. But that is critical information. So that's one of the reasons why Indonesia has been a top priority of mine as far as visiting here and establishing collaborations. So you folks, as graduate students and public health students, are welcome to join this effort. It's a very important one, and I could see that this would be a good way to initiate this collaboration. A strong 10-year effort, I think, could help us make great progress. So with regard to chironex, or the larger chirodropid fatalities, you see the hotspots, other jellyfish. Hawaii has irukandji cases. We also have a few deaths. We don't have chironex, however, in Hawaii. So back in my home in Honolulu, we have beaches on the lee side of the island where the jellyfish come like clockwork 8 to 10 days after each full moon, and they actually swim against a current and swim into these areas that have breaks in the seawall. And my lab and myself go out from midnight to 6 a.m. as they're coming in and collect them by hand. So they also come into the other shores, Hanama Bay, Pokai Bay, Yokohama Bay, all along the lee side, leeward being that the predominant trade winds are coming from this direction, from our northeast. So the mountains of Hawaii, the Ko'olaus, protect this side of the island from wind, and we call that the leeward side. So we have a calendar now to warn the public about times of danger. And we've been taking the data month by month for years. And we have here the annual totals. So what does this look like? I'm also a diver, and I spend time offshore at night scuba diving to observe and film and collect these animals. And we need to push the play button. So here I am 2 miles offshore at 1 a.m. and there is a little box jellyfish. You see that it's white because it's filled with sperm or eggs. These are adults that are getting ready to spawn. So they swim ashore in order to reproduce, and it's called a spawning aggregation. Again, the exist around the world, and we have the two main classes within class, or two main families within class Cupozoa box jellyfish. Again, as a scuba diver, I'm collecting the animals, and I wear typically full face masks for this job so that I'm not stung on the face. Sorry. Here's what a sting looks like. You can see that this is clearly a very dramatic event. The contact of the tentacle with the skin leads to the injection of venom at the site, which causes a searing, burning erythematosis at the site, similar to other envenomations. As a biochemist, I was working happily away in my laboratory, publishing basic biochemical findings as far as the composition of the venom. And then one day I was contacted by the United States Special Operation Command. Combat divers in our Department of Defense had been stung. And this is the Special Forces Operations School in Florida, Key West, Florida. So these folks are United States combat divers that drop out of helicopters at night, and they have a rebreather on. They're doing underwater scuba missions that are 4-hour long, and they're in areas of the tropics. Unfortunately, a number of these very fit young men had career-ending stings, and what that means is that while they were engaged in their training operation, the box jellyfish had gotten into their combat uniforms. They don't wear wetsuits because they'll get overheated. But unfortunately, they had no treatment. So they contacted me to investigate whether I had any treatment that could be useful for their purposes. So then, with that inspiration, I became much more interested in quickly finding solutions that could be used for direct application. So this kind of challenge, as far as why it's fun to be a scientist, comes along where you may be on a particular career track, and my training as a biochemist was basically to find interesting natural mixtures and to solve what are the ingredients of those. But now hearing about real human health suffering, I was fully inspired to try to take my findings and bring them to some conclusion that could be useful in this format. So they funded my work to come to Florida to try to do the kind of field work I was doing in Hawaii to identify what the species in Hawaii were that were causing so much injury to these night scuba divers. So much like I'm here in Indonesia 10 years later, trying to determine what the culprits are here in Indonesia that have caused deaths. I started out leaving Hawaii first, going to Florida. So here we have our vessel offshore, and we have two down lines. Basically, these are lines with lead weights at the end. And then we have a BC that we call a buoyancy control device, and we hook a line from that to the down line. So me as a diver then, a little bit like a puppet on a string attached to the vessel. So this is in the open ocean where the bottom is over 2,000 meters, and we're just in the top 100 feet of that water column. So as we go around, though, the boat is drifting because it's impossible to anchor, and all manner of invertebrates come by. So here's a picture of a box jellyfish I took in such a survey. Beyond the Caribbean, Florida, and Bonaire, I've also done a lot of work in the CMNI, the Micronesia Commonwealth of Northern Mariana Islands, and here is Saipan, and I've done the same sort of drift dive field ecology at night in Saipan. So going out there, we also see oceanic white-tip sharks, and these guys can come by, so that's a little interesting, but we keep doing our work. So this, if we can press the play button, the fellow at the computer, yeah. There we go, yeah. So this is what it looks like, me taking a movie at night in Saipan. The ocean floor is about 800 meters below me, and I'm at about 80 feet below the surface, and this critter comes by, this box jellyfish. Oh, maybe you want to play it again? The breathing is me, not the jellyfish. Okay, so the breathing was me, not the jellyfish. I want to point out that that's the same kind of work that I want to do here in Indonesia. And last Saturday we went out off of Situbondo by a boat. A local fisherman and his father took us out. So Dr. Almanir and I dove into the water and I did some free diving and some basic photography. We didn't find any box jellyfish that night, but I want to go again the 2 more nights while I'm here. Okay, so while we do not know anything about box jellyfish in Indonesian waters, we do know a lot about box jellyfish in some of your neighbors' waters here in the Indo-Pacific. For instance, in the Philippines and in Thailand. So I've done field work in both of those places, and I'm briefly going to talk about that. In Thailand, in the Gulf of Thailand, there's one tiny island which has had more than its share of deaths and severe injuries. In just the last 6 years, there have been over 4 deaths in Koh Samui and far more injuries. We have, with collaborators around the world, been focused on this area, so it's gotten a lot of attention. Australian and Thai researchers have identified all of these different box species being present in the Gulf of Thailand. And again, throughout the world we find these animals, but to focus your attention, this triangle from Philippines across to Thailand down to Australia seems to be their preferred habitat around the world. And as you notice again, Indonesia sits right in the middle of that. And these animals, I should tell you, can be as tiny as this malokingi. The fisherman that we were with described the death in Situbondo as being due to Olayan, and he described it as a very small animal. So we could be looking for something as small as this species from Australia. Now, as far as the Philippines go, we have quite a lot of data, and each of these numbers that I've listed here have over 10 deaths. So in just one tiny little bay up here at Pangasinan, we've had over 100 deaths on average per year. And these are local fisher folk who are out tending their fish traps. And when they pull the fish trap line up, they're not wearing a shirt. And the line can be tangled with jellyfish tentacles. And as it contacts their chest, they get a sting. And that sting can lead to death, and it's often a very rapid death. So the public health needs in the Philippines are profound, and it's well known. It may be that Indonesia doesn't have so many deaths, and that would be good to know, but it may be that it is just below the radar of detection. So public health epidemiologic efforts are absolutely critical to answering that question. So just to give you an idea about the heartbreak involved, here's a story from Mindanao in Philippines, Digos City. A small boy just finished from his elementary school, went to the beach right next to his elementary school, less than a kilometer from the school, and just waiting near the shore was stung. Within 3 hours, he was declared clinically dead at the local clinic. And unfortunately, beyond the danger of the sting is inadequate knowledge as far as the preclinical and clinical care. Here's a mistake in what was reported as far as scientific accuracy. The local knowledge base indicated to the folks writing this news article that vinegar has been confirmed to effectively treat jellyfish stings. I hope you realize as public health students, the word treatment has an important connotation. That means that it can lessen the disease state. And in fact, vinegar doesn't get into the skin. So it is scientifically inaccurate to call vinegar a treatment. That said, vinegar is a good first aid. It prevents things from getting worse, and I'll describe that to you in a little more detail later. But beyond the importance of public health outreach and good surveys, as far as marine surveys, knowing what's going on, is bench research. So I would say that part of the answer to why it's fun to be a scientist is that it's fun to work on teams, and it's fun to work on international collaborative teams. But it's also challenging because we need to engage and recruit members of the team. So for instance, in this room, there might be public health graduate students who could be engaged in epidemiologic research. That is to say, who could send out surveys to coastal communities to see if oboroboros occur there and if they've led to death, and to get more clinical case information. But the other part that public health graduate students could play in this is to communicate effective treatments and to deal with common myths that may actually impede proper management of sting. So in this case, one of the myths, we go backwards. In this case, one of the myths was that vinegar was a treatment and that it would be sufficient to simply put vinegar on the boy, and that's all they did. And in cases in Thailand, we have many case stories where they arrive at hospital and they're just covered with vinegar. Vinegar is not a treatment. Vinegar will only be useful in one step, and that is to first aid, to inactivate stinging cells that haven't discharged on the skin. The next step should have been to immerse the tissue in hot water. These marine venoms are extremely heat sensitive and are inactivated already once they're injected into the skin by hot water immersion. So I hope the takeaway that you bring from this talk for yourselves or your relatives, if they're stung by these serious marine animals, is to first flood the site with vinegar just to clean it of other microscopic stinging cells that could still discharge, and then to soak the site in hot water. Of course, if there's breathing or cardiac issues, they should seek advanced medical care. Okay, so I don't want to get too far into the details of the data of my research path, but as far as answering the question of why it's fun to be a scientist, for me personally, the greatest pleasure is to address puzzles, to find a worthwhile puzzle to focus on, and then to utilize all my education to address that puzzle. So the puzzle that I've been focused on for 19 years is, what causes cardiac arrest after the stings of chironecs and other box jellyfish, the so-called world's deadliest venom? And when I started this work, the conventional hypothesis in the field was that the chironecs venom contained a silver bullet type ion channel blocker. So I started my work with that premise in mind, and actually I found it was quite different from that concept altogether. As far as how this presents, the clinical presentation, the stings present with extreme pain and catecholamine surge features, including anxiety, nausea, sweating. The whip-like lesions, which I've shown you photos of. The death can be rapid. The classical anti-venom treatments that are so helpful in snake envenomation managements don't work. And basically there's an important reason for that. These box jellyfish venoms work far too fast for a neutralizing antibody to be effective. So they are absolutely not been validated in the field use, although they exist. There's a hypertensive, then hypotensive phase of the presentation, and a multi-system cardiovascular event. So to kind of jump way ahead in the talk, but to be sure that I hammer this home and don't leave you with a whole long talk without clarity. As far as buru envenomations and box jellyfish in particular, the best approach is avoidance. And again, this is a call out to public health graduate students here, that in terms of your expertise in reaching to the folks at risk, warnings are the first step. We want to have good signage on beaches in which stings have occurred. I've been to Situbondo, the beaches, and there was no apparent signage anywhere, although deaths have occurred there. So we would like to see avoidance, and simple exposure suits can be used. So simple lycra, the full body kinds of swimwear that many women in Indonesia use already is good. If there is a sting, I have developed these technologies now that are in use by commonly available in the U.S., commercially available as well as abroad, and to the military. So this spray under the registered trademark of Sting No More and Cream. The spray, I should tell you though, is based on the vinegar technology. So if you didn't have this spray, you could use vinegar, simple food vinegar, to again clear the site, the sting site. After that, this highly effective pharmaceutical grade cream is effective. If one didn't have that, then soaking in hot water would be useful. So this is just, summarized here. Do not use cold packs. They simply preserve the venom, and the pressure can release more venom. Do not use pressure immobilization. I looked up on Wikipedia Indonesia, and there's a page on box jellyfish, but that page has many errors on it. And when I translate to English, I could see the Indonesian Wikipedia page is recommending site mobilization, pressure bandage. That has absolutely been disproven over 15 years ago. So I hope that the Indonesia website can be corrected. Do not apply cold fresh water, and do not scrape the sting. If you didn't have vinegar, it could immediately rush to hot water immersion. So another application of my technologies has been with the swimmer Diana Nyad, an American who swam from Havana, Cuba to Florida. Has anybody in the room heard about her swim? If you have, could you raise your hand? Nobody? Okay, so she swam 110 miles, 53 hours without touching the vessel, night and day. I was her diver at night and in the water with her from sunset to sunrise. And here she is after swimming for 53 hours continuously. I'm the one in the white hat. I took this picture of her. But she used this technology as she swam through box jellyfish. So another interesting area, I know that there are biotechnology folks in the room, and another interesting area of opportunity for students in Indonesia are interesting compounds. Now, I've been speaking a lot to the public health students because I hope that they'll join the team with Dr. Almunir to warn local folks. But biotechnology folks can also have a great deal to work with regard to Cnidaria species. Here is shown Physalia physalis that I collected in Florida. They're much larger than the Pacific version. But what we found is that they have novel fluorescent proteins. And I have a fully granted US and international patent on these far near-infrared emitting fluorescent proteins. So this could be a topic of collaboration also between my lab and folks in Jember, in that you have access to lots of the Physalis utriculus here in the Indo-Pacific. What I have found is that this far red emitting fluorescent protein can be used as far as tagging goes. It is what they call in that most desired range as far as to be used for whole body imaging. Near-infrared emitters can be used in deep tissues, and they aren't occluded by the overlaying tissue structure. So this is an area of biotechnology laboratories here that would have HPLC and protein sequencing capacity could certainly join in this collaborative effort. Now I want to go a little bit quickly. I don't have any kind of timekeeping device near me. Can you, is there some way to tell me how many minutes I have left? I don't have a clock in the room. Okay. Be sure and warn me when I'm getting 10 minutes to the end so I can not go over time for you. So I want to walk through the actual path of discovery. So as far as why it's fun to be a scientist, as I mentioned to you, I am motivated by puzzles. And I hope most scientists actually go into science because they never really grow up. They keep that childhood sense of why. And they continuously ask the question in their universe, why? Why this, why that? All right? Now, we get quite an extensive education which answers the question why in a lot of ways. But as you go into the real world, you find actual problems that haven't been solved, where we use previous information to put this together in protocols and approaches to solve currently relevant and important questions. So with regard to why do people die from the box jelly stings, this has been my path. First, I went about to figure out what are the two aspects of a sting. Firstly, there's the physical trauma. Something is piercing through the skin. And then there's the pathophysiologic effects of the venom itself. So the physical trauma required that I did a little bit of basic research into cell biology of the cnidae and the tentacles, and doing some electron microscopy to really identify the structures that are used. The pathophysiology utilized my training in biochemistry as well as bioassay and development of novel animal models. Ultimately, my objective was to attempt to identify the lethal components. You can imagine that if you have a complex natural mixture that has 200 ingredients, that you're trying to answer the question, why did the person die after this exposure? The answer could be that it required just one of those 200 ingredients, or it required a suite of ingredients, maybe 12. So I wanted to answer how many of the constituents are actually most problematic, and is there any way to inhibit those? And ultimately develop an animal model to test therapeutic interventions. Okay, so as far as the physical part of the story goes, here's the tentacle, and here are the individual nidae tubules by electron microscopy. And you see they look like little wires with barbs on them. And when we go to higher magnification, you can see that these tiny spines cover the hollow tubule. This is a cross-section. So it looks a lot like a hypodermic injection needle covered with spines. These spines are mineralized chitin. That is, that they contain the same minerals, in fact, that crab shells have. So the calcium and phosphorus, etc., of a hardened crab shell. So this is what gets injected. Now here's a little movie showing animals we collected in Hawaii, and then on my microscope I attached a high-speed camera. I borrowed a $100,000 camera from Olympus Industrial, and I attached it to my microscope, and I made this film that was used on many documentaries, including National Geographic, where I'm shooting at 33,000 frames a second. That is such a high-speed camera that if you were to film a gun shooting, you could see the bullet frame by frame by frame. So that's how fast this discharge occurs. So the discharge is actually the fastest known biological event. Each one of these stinging cell nidae, and the penetrant ones are nematocysts, has within it the little coiled physical apparatus which, when it's stimulated to discharge, pierces out at bullet speed and pierces through the tissue. So now moving on to the pathophysiology and the biochemistry, I developed a technique that allowed me to isolate intact nidae. This is an electron microscopy cross-section using transmission electron microscopy to show the structural bits, the tubule all coiled up inside of this, as well as what you see in between the structural bits is the venom. And I want you to notice that the venom is dark, and from electron microscopy, it is electron dense. It has the attributes of bacterial storage granules. So this venom is not like snake venom at all. It's basically encapsulated in these very tough nidae that themselves can hold 1,000,000 G force. Once this is opened and discharged, water rushes in and hydrates this highly concentrated gel to form the venom that comes out. So I've developed a technique that allows us to isolate intact nidae from live animals, never frozen, never lyophilized, and then we rupture them in mass with a French press going to 20,000 psi, and then we snap freeze after a hard spin to get rid of the structural bits under liquid nitrogen. And we can compare our method with other methods that have been done previously. And just to show you, in a logarithmic scale, it's about 500 times better than quite a number of the other methods. So it's very important the way in which one isolates the nidae. So this is my own blood, and after applying some of the venom to it, we see even within the first frame, the platelets already aggregated, the red blood cells crenulated, and now they're swelling, and now they're forming ghosts. So the white blood cells have formed foam cells, the platelets have aggregated, and the red blood cells formed ghosts. This is important because this speaks to the pathophysiology. If you look at the venom effects on whole human blood, you can make a lot of determinations about what's going on in the body. So I won't belabor this because it isn't precisely your reason for being here today. But another part of why it's fun to be a scientist is to use your data and to use multiple approaches in order to develop theoretical models. So this is my current theoretical model of what goes on in box jellyfish sting. So what we found was that actually the venom, when we fractionate it, the one small fraction was able in and of itself to cause death, the same as the whole venom in mice and piglets. So we went to look at that more carefully, and we found that it was a porin, a pore-forming protein. And this is an electron micrograph of a red blood cell surface, and you see that these porins have formed these rivet-like holes. This is very similar to what happens after blood exposure to anthralysin O and streptolysin O. So pathogenic bacteria release these porins, pore-forming agents, which actually confound the host system. So when you have a bacterial infection, blood macrophages will engulf the bacteria and try to destroy it. In the case of anthrax and strep bacteria, they can produce large amounts of these porins, which, going forward, sorry, these porins, which then insert into not just red blood cell, but white blood cell and platelets, forming these holes. And basically it's a get out of jail free card, if you will. So the bacteria destroys the macrophage that's engulfing it. This occurs, as I mentioned, to platelets and to white blood cells, and results in this whole constellation. So this is basically the short form of my 19-year adventure into trying to answer my original puzzle. We found at the end of the day that it's simply this highly conserved porin that's present in all the cubozoa that causes this rapid death, as well as, so there's a release of potassium from red blood cells, histamine, cytokines, catecholamines. At high enough doses, a massive uptick in plasma potassium will lead to death on the spot due to what's called hyperkalemia. How many people in the room have heard of the Nernst equation? The Nernst equation. Okay, put that on your homework, write that down. Everyone in this room should know the Nernst equation. Why that's important is because it shows that the level of ions inside the cell versus outside the cell determines the ability of the cell to survive. So in cases of high external potassium, the cardiomyocytes cannot beat. So transient hyperkalemia can lead to death, but it isn't cardiac damage. So that's another important point that my work has made clear. As far as another sort of call for collaboration with biotechnology students in the room, with regard to Indonesian species, there is a great deal of discovery that can be made using biochemical separation, and this is my work with the box jelly venom to fractionate and to do mass spectrometry to identify novel compounds. These have often been used for drug discovery for treatment of other disorders. So from another general structural schematic of what goes on, these cnidae penetrate through the tissue and release the venom, which affects the dermal neurons with direct pain to cause this burning pain by the porins inserting into the neurons, causing holes, much as heat burning would do, and these other effects. Okay, so this is a paper reviewing what we found as far as hyperkalemia, that the death was simply due to a massive increase of potassium, not from the proposed silver bullet ion channel blocker. And this is work in my lab with a mouse, and we do simultaneous echocardiography as well as EKG reading, and we can see this is left ventricular contraction. As the plasma potassium rises, the blood, the cardiomyocyte is no longer able to contract. And so you have pulseless electrical activity. There's no more contraction, so no more pulse. Yet by EKG, you can still see some electrical activity until the animal perishes due to inadequate oxygenation. And this is another part of the fun part for me, is putting together these models. So this is just a list of everything that I found that occurs upon exposure to the porin. And then at high levels of exposure, we go down this red pathway to death. We've also taken this model now to piglets, and we are measuring all of these different attributes as well as many others, and then testing different clinical therapeutics to save a lethally injected animal. But just to jump again now to sort of a bigger picture, what would help in a first aid situation? On Wikipedia and WikiHow, you'll find you should scrape it, you should add vinegar, you should add ice, you should add shaving cream, you should have someone urinate on it, or you should use one of these items. So you find a lot of, in the public health space, a lot of misinformation. We know that scraping it in any way makes things far worse. Vinegar, yes. Ice, no. Shaving cream, no. This, no. So basically vinegar and the hot water follow-up, or the other technology that I described previously. So for folks in this room who might be interested in working on this as a public health project, this would be a nice way to organize your thinking. In terms of developing data to address evidence-based first aid in Indonesia, part of the reason why I'm here is to actually catch animals, live animals, local animals in Indonesian waters, and to do various assays and to test experimentally the effects of different potential ways to reduce poor outcomes. And I'm not going to go through this in great detail, but I would encourage you to contact Dr. Munawir if this interests you and you might like to talk about it further. But one of the things that I've been demonstrating in his lab this past week are these different assays. One of them is a tentacle solution assay, in which we add different solutions to a tentacle on a glass slide. But as far as the survey of all the techniques that have been used in the literature and the pros and the cons, again, I'm not going to go into this in great detail, but I would encourage you to contact Dr. Munawir if you'd like to work on this. So again, as part of being a scientist, we want to objectively go through everything that's been published with regard to a particular research question of interest, and to organize the thinking in terms of what are the advantages or disadvantages of different approaches. So as a conclusion of that work, I've recently published this paper, and I would encourage you to read that if that interests you. But in a quick snapshot, what we've done is to take whole human blood or washed red blood cells and combine them with low melting point agarose, and we form an optically clear gel in which the red blood cells are still alive and quite healthy. To that, we can apply a tentacle, and the tentacle will spontaneously sting this gel. We can then do various different assays. So here's just pulling off the tentacle. The outline is where the tentacle had been. If we rinse with seawater, we see all the white area here is where hemolysis had occurred. So here, hemolysis was simply limited to the site of the tentacle, whereas when we flooded with seawater, the hemolysis spread. There were more sites that the Nidae injected the venom and caused red blood cell damage and destruction. If we applied the vinegar, there was far less than in the seawater, far less white spots. If we apply fresh water, there's more than either the pulling off or the vinegar. If we applied ethanol, again, more. And if we scrape off, the absolute worst case. As you're scraping the tentacle down the auger or the skin, in the case of the potential child being stung, you're causing the nidate to slough off again and again, and the pressure is causing them to sting and inject the venom. So here's an experimental design that addresses what's on WikiHow. And this results in scientifically rigorous, what we call evidence-based outcomes. So we can say, people have sat in their armchairs and thought, well, we won't use vinegar, we'll use seawater. Seawater couldn't cause any problem. The animals live in seawater. And in fact, seawater just protects the nidate and they can continue to sting. Okay, and then heat we find is far better. So this is, on the y-axis is the amount of hemolysis, which at a certain level equals death. And here is time. So if we treat with various options, seawater rinse, no scraping, here, vinegar rinse, no scraping, vinegar rinse with scraping, basically no difference because vinegar just inhibits everything from the start, versus hot or cold treatment after the fact. So if we, in almost any event, if we'll add heat immediately, we have the least hemolysis. And this is how the auger plates look. So we apply the tentacles and we either put them on a hot pack or cold pack. And the more clear zone you see, that equals more hemolysis and in a certain level death. So you can see that the hot packs reduce the development of a hemolytic zone, whereas the cold packs did not. Here's my technology, the Sting No More cream. Without hot or cold, it basically stopped all hemolysis. Okay, so just to wrap up, as far as the current and proposed international collaborative efforts, I am here to work on building international collaboration to collect live cubozoa and to train local students in the direct assays in vitro as well as in vivo, and to investigate the therapeutic, to have local folks investigate these therapeutic options with their own animals in their own labs. I won't go into this, but this is what's involved in the development of the technology. And terima kasih banyak.
Thank you, Profesor Angel. Thanks it. Sekarang kita buka sesi pertanyaan ya, question. Silahkan yang tertarik ya, terutama yang paling akhir tadi ya. Jadi ada saya yang terakhir. Jadi kalian terbuka kesempatan untuk mengikuti beliau ya. Bukannya di sini aja. Insya Allah beliau siap ya nanti menerima kalau kepingin ke Amerika ya. Ya. Silahkan pertanyaan mungkin. Question ya, satu lagi. Ya, dua. Oke, sementara dua dulu ya. Silakan. In English ya.
Terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Good morning or good evening. Prof. Angel Eni Yana Gihara Pihadi. My name is Yuni Puspitasari. Saya sekolah di saya jurusan S2 Ilmu Kesehatan Masyarakat. Okay, number one, how do you scientific transfer of Mary jellyfish from Australia to the island of Java? There are cases in Python Probolinggo affecting thousands of jellyfish and disrupt fishing and power plant their way out. It is number one. And number two, wherever the factor may be to ekstrim cuaca ekstrim di Australia, campuran ya, nggak apa-apa ya, yang penting berusaha. Tepuk tangannya mana? Bahasa Indonesianya begini. Bahasa Indonesia ya, ngomongnya gak ngerti bahasa Inggris. Pak, bahasa Indonesianya begini ya Pak. Dulu ada kasus di Python itu ribuan ubur-ubur datang ke Probolinggo. Kan ada yang translate tuh Bapak. Ya. Daripada saya salah bahasa Inggrisnya, malah gak ngerti nanti profesor. Oke. Ubur-ubur itu datang ribuan. Apakah itu faktor cuaca ekstrim yang ada di Australia, dingin, mengakibatkan ubur-ubur itu migrasi ke Pulau Jawa? Dan bagaimana cara solusinya untuk bisa segera mengusirnya? Karena mengganggu nelayan dan pembangkit listrik tenaga uap untuk kinerja PLTU-nya pada saat itu. Itu yang pertama. Yang kedua, Bapak, bagaimana Prof, Bapak, iya jangan bikin. Terus habis itu, yang kedua Pak, jika nelayan itu terkena ubur-ubur pada saat itu juga, bagaimana penanganan pertama yang dilakukan masyarakat awam atau para nelayan untuk mengantisipasi terserangnya racun ubur-ubur pada saat itu. Itu saja mungkin yang paling mendasar. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. Thank you, Miss. Tepuk tangannya mana?
Langsung aja ya biar nggak lupa nanti. Profesornya biar nggak lupa ya. One question is she asked you about blooming jellyfish in we have one area and near Probolinggo, Python. From where and why?
Okay. It isn't accurate for us to call the numbers of box jellyfish as a bloom. So within the Cnidaria there are different classes. There are the Cubozoa, the Scyphozoa, and the Hydrozoa. So around the world, what's caused, I heard at one point you're talking about the nuclear power plants. That's a story from the Philippines, just from the Philippines. I don't know anywhere else in the world that has occurred. But that wasn't a box jellyfish, that was from class Scyphozoa. So Scyphozoa actually form huge blooms. And I have also done work in Ireland because these massive clouds that are kilometers wide go up the North Sea, and they're coming across the salmon fisheries and wiping out these pens with tens of millions of dollars of salmon. So with regard to Situbondo, I have zero evidence that there's any blooming going on there. I was there the other night and I saw quite a lot of ctenophores, which are gelatinous little animals that have this sort of strobulating rainbow, but they're not jellies. But with regard to the field ecology and the life cycle of box jellyfish, in Hawaii we have them coming ashore 8 to 10 days after the full moon. One particular species does that, only one out of the 40. And on those nights, we can have thousands of box jellies. It's not a bloom, it's a spawning aggregation. So they're purposefully swimming up from the deep ocean off Hawaii and coming to this coastal zone, and they reproduce and then die. So that, I hope, is the answer to one of your questions. And go ahead with the next.
Oke, jelas ya. Jadi bloomingnya itu karena di situ aja ya, karena perubahan cuaca bisa juga ya, dia naik ke atas ya. Dan yang blooming tadi itu bukan yang berbahaya ya. Jadi yang di Probolinggo itu bukan pork jellyfish ya, tetapi dari spesies yang lain ya, bisa sibojoa atau kubozoa ya. Kita sudah explore ya semalaman 2 hari yang lalu ya. Itu kita tidak menemukan jellyfish, apa mungkin waktunya kurang tepat. Kalau di Hawaii itu setelah full moon ya, dari bulan-bulan mati itu ya, baru kita ke sana malam hari. Makanya hari ini nanti kita akan diving lagi ya, 2 hari. Kalau barangkali ada mahasiswa yang mau ikut, gratis ya. Ya, di situ Bondo ya. Question number 2 is about the issue about the first aid for fishermen if they get stung by jellyfish in this in the sea.
What is the first aid for jellyfish among fishermen? We spoke with our local fisherman, Mr. Brody, and he knew about vinegar. Also, he said sometimes they use fresh orange juice or coconut to drink or to put on. As far as evidence-based research goes throughout the world, the best thing to do with all the species that I've looked at in all the classes is to first flood the site with vinegar, proper household food-grade vinegar. After that, soak the area in hot water, 45 degrees for 45 minutes. If the person is presenting with any breathing or cardiac issues, to get them to hospital as quickly as possible for that oxygen by mask or CPR, cardiovascular support. Also, by flooding with IV saline, you can assist in reduction of plasma potassium if that's already begun. So that would be a safe and effective intervention. If you're interested in absolute clinical care protocol from a physician, I can put you in contact with a physician on our team, a dive medical officer from the Special Operation Command. They can send you a particular clinical care schematic, flow diagram of what to do.
Baik, ya sudah jelas ya, masyarakat itu sudah tahu, cuma memang ada beberapa yang ternyata itu kalau diteliti tidak efektif ya. Beliau tadi menyarankan ya, pertama menggunakan cuka ya, kemudian habis itu disiram pakai air hangat ya, selama 45 menit ya. Nah, itu pertolongan pertama. Untuk apa? Untuk mengurangi agar racunnya itu tidak masuk banyak sekali, dan yang tentakel yang belum aktif itu tidak lepas ya. Itu saya kira itu. Dan kemudian kalau memang ada tanda-tanda yang gawat ya, kan jantung apa sesak, harus segera dilakukan pertolongan. Memang lebih baik ya, begitu kejadian kalau ada tandanya langsung aja dibawa puskesmas ya. Ya, saya kira itu. Silakan question number 2.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for your lecture and opportunity. Prof Angel, you look so beautiful with that scarf. I like very much. I have question. In your lecture, you mentioned about lethal component. I'm interested in that phenomenon. there is any possibility we can use that phenom for protection in plant, like BT crop or something else, like because I'm working in plant protection, maybe any like publication or something else, you can mention, I don't know what kind phenom in here, but I'm interested, we can use or not this phenom for plant protection, like transformation or something else like that. Thank you.
That's a fascinating question, and it's part of the answer of why it's fun to be a scientist. You're asking me a question I've never heard before, and I've never even thought of before. So I really have to take some time and think about that. I've never been asked about plant protection application from the venom constituents. I think I would need to know a little bit more about the particular protection classes you're investigating. Is this to kill a potential arthropod insects, or?
Yes, in my mind, I try to thinking in different way. Okay, we can use this phenom for biotechnology, like we know before PT. Bacillus thuringiensis. Yes. This Bacillus thuringiensis produce crystal protein, and this effective to kill larvae and insect. And this technology, America use leading in this technology. And in my mind, because the title is fun to be scientist,
right.
And I try to while thinking. Maybe there is any possibility we can use this phenom for protection, plant protection. Example, we try to identify the protein, and after that we try to isolate the gene, regulate that phenom, and we use for transformation in, example, cotton plant or something else like that. I think this is very interesting.
So I'm going to ask Dr. Munawir to get your email and really consider your question properly. But off the top of my head, in the Scyphozoa, such as Nomura, some of these more primitive jellies, they use non-penetrant naidae called tychosis. And it's very interesting. They use a strategy that's a lot like a spider. So a spider makes a silk web, and it puts on it some bits of chemicals which are toxic to the insect. And when the insect flies into the web, he's physically stuck there. There's also an adhesive material, and there's also a bioactive material. So the insect has physical and biochemical effects of being trapped. Interestingly, these large jellies such as Nomura don't sting their prey because they're eating tiny microscopic copepods, which are very similar to terrestrial insects being in the arthropoda class. So within that class, this is a marine environment, but the Nomura tychosis shoot out a microscopic net. It's a tiny net which has bioadhesive on it, and it traps the copepod, so it actually enmeshes these tiny microscopic animals. So there may be some area there to explore from a biotechnology point of view. It sounds to me like it would be a fascinating grant proposal to write. I don't have an answer, but it is an interesting question, and that usually means that this is a great opportunity for someone fresh in the field to explore and to write a proposal to do this kind of work.
Oke, saya kira dah ya dikasih gambaran tadi ya, walaupun belum ada jawaban, justru itu menjadi tantangan ya untuk meneliti lebih lanjut. Ya silakan, mungkin ada lagi. Ya,
Lagi mungkin yang lain ada lagi? Satu lagi? Oke, nggak apa-apa, satu. Silakan.
Thank you, Professor, for the opportunity. My question is, what is the biggest challenge when you do your research, and what is the solution? Thank you.
The biggest challenge. Well, I can't say just one. Certainly funding. Without funding, it's over. You can't be a researcher. You must have some grant support or institutional support. So the answer to that, I suppose, is to be able to write good grants, and that really requires that you have a good, compelling question, that you have a well-organized strategy, that you have a compelling team and environment, and that you have a good work plan. So, but beyond that, the real big problem is that biology and the natural world is a very complex one, and it's easy for you to get lost and basically end up with no answer. So the strategy that I've followed over the last 19 years really started when I was your age, and that is to try to take as many courses as I could, to try to master as much of the basic scientific literature as I could. So basic biology, basic chemistry, biochemistry, microbiology, immunology, I loved it all, I studied it all, I worked as hard as I could to memorize and learn everything I could. So part of, I think, the biggest problem, it's either going to be not having funding to do the research or not having the capacity to do the research. So the capacity to do research is something which starts right now in this room at your age, and that is just being dedicated to learning. But it doesn't stop at any age. You must continuously have that devotion to learning. But those, I think, are the two big issues. Thank you.
Ya, ada lagi? Ya, silakan. Tolong anu mic-nya.
Thank you, Professor. I want to have one question. When we have get a string from tentacle of jellyfish, why we cannot use alcohol and cold water? Thank you.
You can use alcohol and cold water, but you will not be helping the situation. So the problem with alcohol is that it causes more discharge, and the discharge goes, the venom, into the skin, so you make things worse. Cold also doesn't improve things over the long 18-hour time course. Application of a cold pack for a muscle sprain is basically not harmful. But if you're applying a cold pack in the event of an active venom that is destroying tissue, you will not remove the venom from the tissue. You simply slow it down temporarily. So while the person may be at the lifeguard stand or wherever they are, in the fisherman's boat, if they put the ice on it, temporarily it slows down the pain, but within hours that venom is going to start working again, and at that point it will work very fast because it's been preserved by the cold. So neither alcohol nor application of cold reduce the long-term hemolysis. So the paper that I showed there has time courses where we've gone out 24 hours. Some of the papers that have been published in the literature that claimed that ice packs was useful only followed the person for an hour or 2 hours. So you really must follow the actual clinical case for at least 24 hours. But better yet is to design good experimental models so you can do statistically powered experiments. So this blood auger assay that I've described allows you to do N of 12 of all of these. So if this is something that's of interest, you might want to contact Dr. Munawir and see for yourself in the laboratory. But we've compared all of these different potential responses to a sting, and we've reported on how that affects the ultimate level of hemolysis.
Baik, saya kira cukup ya. Sudah jam 10. The time is over. Terima kasih atas kedatangan saudara-saudara sekalian, memanfaatkan waktu ini. Mudah-mudahan nanti bermanfaat dan sesuai dengan tadi dari Pak Isa ya. Jadi nanti akan ada kuliah-kuliah lagi ya dari tamu kita. Untuk apa? Agar kita lebih bersemangat ya, sesuai dengan tujuannya. Jadi kuliah di sini ini bukan hanya untuk mencari gelar ya. Ya, saya kira itu. Terima kasih dari saya. Thank you, Professor Angel, for your lecture and answering question. Maybe it is important for my student, graduate student, to get motivation, maybe to be scientist next. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you for Prof. Angela for your lecture. It's really nice lecture, and your lecture has been make our mind is open, and we have to motivate us to search and think more and more for something new, something big, and something useful. Thank you. And also thanks to our moderators today, Dr. Al Munawir, which was guided us in this general lecture. And the last agenda is closing. To close our program, let us recite Hamdalah together. Alhamdulillahirabbil alamin. Finally, from the deep of my heart, I do apologize for any mistake I did in presenting this event today. So thank you very much for your attention. Good luck and keep fun being a scientist. Assalamualaikum warahmatullahi wabarakatuh.
Cek, cek.
Ya, saat ini saya ada di Kuliah Umum di Gedung Surahman di program Pasca Sarjana Universitas Jember dengan tema Why It's So Fun to Be a Scientist. Saya dengan Profesor Angel Anna Yanigara PhD dari Hawaii, USA. Sebentar lagi kita akan wawancara sama Profesor Angel. Ditunggu sebentar lagi nggih. Right here. Stop. Right here. Hello, my name is Adele. I'm sorry if my English not speak well. Okay. If I'm sorry if my speak not well. Okay. Okay. Professor Angel Anna Yanigara PhD. For the first time, I know that you are also a speaker at medical faculty. Is that right?
Yes. Yes, I'm a faculty member at the University of Hawaii, Tropical Medical Faculty.
My question is, I know that you are also a speaker at medical faculty. Before, right?
Oh yes, I was the invited speaker. Yes.
How long you have been here?
Since November 2nd.
And then back to theme, why choose the theme? Why it's so fun to be a scientist? Can you look at camera?
Sorry.
Back to the theme, why you choose the theme? Why it's so fun to be a scientist?
The scientific career is certainly a very interesting one. I never imagined 19 years ago that it would bring me to Indonesia, and I'm very honored to be here. But I would say that science certainly can lead you to very interesting places, and it's a very good career. So I encourage young people to work hard and master the basic fundamentals, biochemistry, chemistry, biology. To work hard in school actually can lead to such an important career that you can address human health issues such as deaths in Indonesia due to oboro-boro and olayan stings.
And why you choose the research about the jellyfish?
That's a really good question. I didn't choose this research. They actually chose me. I was stung myself in 1997, and I almost died in Hawaii. So I was swimming, my own experience. So after being stung, I almost drowned, and I became very curious as to what's in the venom. So that's what began my research. Yes, actually being stung.
And this it's important for me to ask why you far away to come here and why you choose UNED.
Actually, it is Dr. Munawir who has chose me. So he's been very interested in investigating box jellyfish in Indonesia. So he's a great leader here, and it's an extremely important area of the Indo-Pacific. So it's thanks to Dr. Munawir that I am here.
And after this, do you have a plan for has a cooperation with UNED?
Oh yes, we have many plans for continued collaboration. Yes, and I'd like to invite Dr. Munawir to my laboratory in Hawaii. Yes, so that he can see all the things that we're doing there, and to bring some of those technologies to Indonesia, to Jember.
I'm sorry, it's out of topic, Miss. When I meet you for the first, you didn't wear a scarf, right? Okay. Why you wear that for now?
Well, I've been in other parts of the Muslim world, and it's a cultural courtesy, the hijab. So I wish to have a courtesy. I have a great respect for the culture.
I asked about why your opinion about Muslim in here.
Oh, this is a very important question in the world right now, especially in the United States. There's so much drama about Muslim. I think it's important for international scientists to work together. It will show that in reality we are all one people on this earth. We are one race. It's a human race, and we can work together, and we must work together.
Okay, that's my out of topic, right? And now your impression about UNED and Jember. Your impression over here.
I'm very impressed. Dr. Munawir's students in the laboratory have worked very hard. I've been a very demanding principal investigator, teaching them basic research, but they have kept up with me and they never became discouraged. So I think the students here have an extremely deep work ethic, and they have a very open mind, and they're very curious students and dedicated students. So I can see why Indonesia is such a powerhouse in the Indo-Pacific.
Thank you for impression, all right? Okay, now we have to glad to have a photo together, all right? All right. All right, thank you. Thank you for the interview. Next to meet you, okay?
Well, I think hijab is important.
Yes.
Cultural solidarity.
Yeah.
Especially for America.
Maybe you are all respect for the first time, yeah. But you're so beautiful. Oh, you're so beautiful. You're so beautiful. Thank you, sir. Terima kasih, Bapak. Thank you. Okay. Oke, tadi kita sudah berwawancara sama Profesor Angel dari Hawaii. Kita udah tahu bahwa dia impressionnya dari buat UNED itu di UNED attitude murid-murid attitude di UNED itu bagus sekali. Jadi dia senang buat bekerja di sini. Selanjutnya mungkin buat Fakultas Kedokteran sama Fakultas Kesehatan Masyarakat juga mau diundang ke Hawaii. Cukup sekian dari saya, Adel, Mas Makmun, Mas Dian, dan Mas Is melaporkan dari Gedung Pasca Sarjana.